Hi, my name is Andrew Olson. I work on the Gramene project, primarily on data integration and indexing for our API at data.gramene.org and on visualizations for the search.gramene.org user interface. In this webinar, I will outline the roles of Gramene's new search database and interfaces, walk you through the user interface, and give you a preview of some new features we're developing. The Gramene project encompasses a set of best-in-class software tools that specialize in subsets of functionality. Genomes, gene annotations, variation data, and comparative genomics reside in the Ensemble infrastructure. We collaborate with the Ensemble Plants team at the EBI for each release. The Reactome provides pathways and metabolic networks, and we collaborate with the EBI Atlas project to host curated baseline and differential gene expression data for several species within Gramene and other species. These tools exist in what you can picture as siloed databases, services, and user interfaces. In order to better integrate these data, we have built a data warehouse, a programmatic interface, and a user interface designed to make them searchable together. Some goals of the search interface. First of all, we want to be able to search all of Gramene's data from these various repositories and silos. Currently, we are focusing on genes, and we will be also adding phenotype data and things linked to variations that have not been indexed yet. We want this search interface to allow you to easily perform powerful expressive searches, and we want to provide useful summaries and visualizations of the result set, as well as links to specialized tools. And above all, we want this to be fast. So I will now attempt a live demo of the site. So this is the, let me clear this, hold on. The front page of the search interface, and it offers links to other tools within Gramene, as well as a feed of the latest news from our blog. And but I want to draw your attention to the search bar at the top. On the right-hand side, you have what I'll call the search status area, and it shows the total number of genes and genomes that we have indexed in Gramene. If you click on this, you can define a subset of species that you want to focus your search on. So you can pick the species from a list. It provides a list, and you can select, let's choose Oryza sativa japonica, sorghum bicolor, Arabidopsis thaliana, Vitis vinifera, which is grape. And when you return to this screen, you can see that the counts have updated. So you're now only going to be searching within 4 genomes, and this is the total number of genes in those 4 genomes. But for now, let's revert to showing all species. Next to this search status area is a little question mark, which provides a brief guide to searching the site. And basically, you just start typing whatever it is you want to search for. So let's suppose you want to search for genes involved in jasmonic acid signaling. So if you start typing, you'll see that it provides suggestions for possible search filters, and these are grouped by category, and they also contain a count next to each possible suggestion, providing the number of genes that match that filter. Also, the top scoring suggestions in each category come up to the percolate up to the top, so provide a convenient access. So since we want a jasmonic acid, let's keep typing a little more, and you can see that's now been brought up to the top. So if I click on jasmonic acid signaling, it will apply this search filter, and it updates the count. You can now see that there are 81 genes across 33 genomes that have been annotated through the plant reactome for jasmonic acid signaling. That suggestion that we selected has now appeared below the search box as a filter. You can also cancel the filter and clear it back to the original state. But below that, I'll introduce what I call the taxonomic distribution of your search results. So on the left-hand side is a species tree that is organized according to the NCBI taxonomy. And to the right of each species, you see the count for the number of genes in the result set for that species. And to the right of that are these multicolored bands, which represent a linear view of each genome. The colors alternate, will cycle through the colors where each color represents a distinct chromosome. And these gray portions represent unanchored pieces of chromosomes. And so we do not display the location of genes in these unanchored pieces so that you don't get, it doesn't suggest the location of a gene where we don't know the location yet. And now below this, as you scroll down, you can see a more traditional list-based view of the search results. By default, we put Arabidopsis genes at the top of this list because they tend to be the best annotated because of its status as a model species that's been studied for many years. And when there is a curated description available at TARE, we'll also display that here to provide a convenient overview of what's known about this gene. In other rows of the table where it's not an Arabidopsis gene, you will see a model species homologue, which we have identified as the closest well-annotated homologue to this gene in Arabidopsis. It can also help when a gene doesn't have a known description to refer over to this model species homologue. Now, underneath each gene in the search results, you have location, homology, and cross-references. So let's click on the location tab and see what that offers. So what it loads is a lightweight genome browser centered on this gene, and what you see is the transcripts that have been annotated. This allows you to drag around, and something convenient you can do is you position this cursor over a spot in the gene structure, and you can click here to zoom in or jump directly to 100 base pair resolution, and you can see the actual nucleotide. So you can see that this splice junction has an AG acceptor site, and you can identify that this red region, which corresponds to the coding sequence, starts with an ATG start codon. You can also zoom around. You can jump back out to this resolution, zoom out further, and then of course you can reset to the original view. But let's suppose you're more interested to see some more details about the rice gene. So let's click on this location view, and you can see this version in rice has two isoforms, but you want more information, you can click through to the Gramene Ensemble site. And this is the gene page on the Gramene Ensemble site, which, among other things, on the left-hand side, you can click gene expression. I just wanted to show off the EBI Atlas widget, which displays gene expression levels in various tissues. We plan to integrate this widget into the result list, so you don't have to click through all the time and get out of the context of your current search. So another type of search you might do is given the name of a gene. Like, so you know that this gene is called RFL1, for example. And so for this gene, it shows that there are only 9 genes in 2 genomes that have the name RFL1 assigned to them. And 8 of them are in Brassica rapa, and 1 of them is in Arabidopsis. We have a technical problem?
Yeah, I don't know what happened. Hold off for a second. It looks like it's restoring video. This has never happened before. Says it's still streaming. What?
Okay, so let's look at the Arabidopsis gene and try the homology tab. So the homology tab will load the comparative gene tree for this gene. And as you can see, this gene tree is much larger than the search results suggest, with just 9 genes that are RFL1. But in fact, this gene tree has over 1,000 genes in it. So on the left-hand side, you see the tree of the gene tree expanded to display this gene that you selected, RFL1 in Arabidopsis. And on the right-hand side is a compact representation of the multiple sequence alignment that was used to generate this gene tree, where a vertical line indicates positions in the multiple sequence alignment where this gene's protein occurs in that multiple sequence alignment, and the various colors that you can interact with also are annotated domains, the positions of annotated interpro domains. So for example, this green domain is a leucine rich repeat domain, L domain like, and you can see that 86% of the genes in the gene tree share this domain. And another gene here, an NBARC domain, is shared by 92% of the genes in the tree, and I didn't click on one that was in this row, but in a collapsed node here, you can see that in the subtree you also get stats. All 21 genes in this subtree have that domain annotated. So also within the gene tree you have these red and blue internal nodes, and if you click on one of those you can see some more information about it. This was a duplication event, and you can also click a blue node, you see that was a speciation event. But if you pick pick this leaf node, you also have the ability to display, it'll automatically expand to show the paralogs of this gene. So now the gene tree has opened up to display all of the Arabidopsis genes in the gene tree. This can get a bit unwieldy when you're just interested in maybe rice and you want to compare it to the Arabidopsis gene family. So you can filter it by genome, as I showed earlier. But first I wanted to show you these links at the bottom. These links will actually alter the search state of the search. So currently the search is just for this gene RFL1, and if you click Show All Homologs, it'll show all 1114 homologs in the search result. And so if you scroll back up to the top, you can see the distribution of these homologs across all of the species. And what you can see from the numbers here, and just by eye in the visualization, is that there are far more genes occurring in this part of the species tree in dicots than in the monocots. So let's focus in on just 2 species, Oryza sativa japonica and Arabidopsis thaliana. And so what that actually does is it provides sort of a superpowered filter, which is behind the scenes filtering your search results automatically to just the species that you're interested in, but it also has effects on the genome-wide distribution view and on the homology view. So what it did effectively was prune this species tree of any branches that don't lead to your species of interest. And if you expand the homology view again, you'll see the same effect here, where the gene tree is redrawn with these duplication events and speciation events that don't lead to an Arabidopsis or a rice gene as collapsed. And this makes it much more tractable to look at the relationships between a pair of species. So now if we take this gene and show the paralogs, you can see that it's much easier to see what has happened here. And we can also expand this node to see so there's a couple of rice genes up here, and this node has 8 rice genes in it. And so what you can see is these rice genes obviously had an expansion of duplications in their history, but the duplication events in the ancestors of the Arabidopsis genes were much more prolific. So I want to return to the demo is now over and discuss some of the features that are in development. So one of the things that we've heard a lot of feedback from members of our group who've started to use this tool is they'd like to be able to download the search results. Some of these things are more trivial to implement than others. Also, sequence searching by sequence is not something that's particularly lends itself well to the interface that we've designed. So, but we do have the BLAST interfaces at Gramene. So we want to be able to link from BLAST results to the Gramene search interface so that you can continue to explore the results in the context of all the other information that we've been integrating. That interface for selecting genomes of interest is going to be more important to go beyond just a list, especially in the next years, because we'll be expanding to over 100 genomes, likely. So some ideas we have there is to allow the species tree to guide the selection of genomes. Also, we could color code various clades so it's easier to identify the monocots, the dicots, or highlight. We might want to highlight model species or crop genomes. Also, the customizable filter logic would be nice because right now, every time you do a query, the default behavior is that filters that are in the same category are combined with an OR. So, and filters between categories are combined with AND logic. This tends to work in general, but it might be nice in practice to offer a more sophisticated interface for customizing your search, customizing the logic of that filter. We also intend to index the Atlas expression data for search, as well as provide the little widgets to display gene expression data in the results list. And also the plant reactome is undergoing another round of improvements, and we expect that there's going to be some cool new viewers based on a graph database that they have behind the scenes. And another feature that I'll show you more about that we're working on, several sites have such a tool, a gene family neighborhood conservation viewer. So we organized 2 internal hackathons among developers in our group to develop our own version. The outcome of the first hackathon was this working phenotype. And so what you see is in the top row is a gene that was a query gene, and it appears in the center of this visualization. The orthologs of this gene are arranged below it, and you can see the gene neighborhood of each of those orthologs. The color scheme is defined by that neighborhood of the query gene, and each gene tree is assigned a distinct color in that neighborhood. And these gene trees, since they occur, these gene families are spread across all of the orthologs. You have these colors that you can then track visually where certain gene families may be conserved across evolutionary history. You can also adjust the size of the neighborhood from the current default of 5 up to, say, 20 genes on either side. So in our second hackathon, we focused on making improvements to this, such as better, to make it more useful and easily interpreted. So we're using different shapes to distinguish the biotypes. So we have protein coding genes, non-coding genes, and pseudogenes that get special treatment so you can distinguish them in the view. We also use shading in the center column to show the percent identity relative to the query gene. And we added arrows on either side of the neighborhood to indicate when a whole neighborhood was flipped in order to keep the center genes in alignment. We also are able to given the set of genes that come out of the query for the neighborhoods of all these orthologs, count features that occur in this result set, and you can filter and highlight. You can highlight the genes by these properties. So I will do another demo here. So here you have a quick look at one gene, and you can see as you mouse over things, it gives you information about the gene, its ID, a name, the taxonomy of this gene, the region that it occurs in, its biotype, and as you scroll down, you can see like this percent identity is also moving along with that. Other things you can, so you can adjust the scale here. So now we have a 10 gene neighborhood on either side, and you can see which domains are popular among genes in this view. You can click on this, so highlight these domains. You can also clear the selection. There's also roots. So by roots, I mean the root and taxonomy node of a gene tree. And so if you wanted to say, there are 43 genes that have a root taxon ID that's a rice. So these are rice-specific genes, things like that. So the next step hang on. Yeah, so one of the things we want to do is integrate this into the multiple sequence alignment in the homology tab of the results of the gene search. And so finally, I just want to acknowledge the group. It's distributed across various sites, and I work in Dorian Ware's group. And until recently, Joe Mulvaney deserves a lot of credit for this, but he's moved on in his career. And so in light of that, we are hiring. So this could be your view. That's all.